In this lecture, we're going to discuss detecting and identifying microorganisms. At the end of the lecture, you should be able to identify the advantages and disadvantages of using molecular-based methods compared to traditional culture-based methods, to explain the value of controls, in particular amplification controls, in ensuring the reliability of results, and compare and contrast the molecular methods that are used to type bacterial strains in epidemiological investigations. So there are specific organisms that have been targeted for molecular-based testing. And the reason that some microorganisms have been targeted are because some bacteria are difficult or very time-consuming to isolate. For example, Mycobacterium tuberculosis takes 2 to 3 weeks to grow, and other species of Mycobacterium take even longer. Some organisms are very hazardous, such as histoplasma and coccidioides, so you really don't want to grow those organisms up in culture because they're highly infectious. Also, some microorganisms don't have reliable testing methods, such as some of the viral infections like HIV and hepatitis C virus. And also, there are some high volume tests done in your typical microbiology laboratory, and making those molecular based just makes them done much more rapidly. So these are your strep pyogenes, Neisseria gonorrhea, and Chlamydia trachomatis. So why do we want to use molecular based testing in clinical microbiology? Well, we want to rapidly identify microorganisms that are causing disease. We want to detect and analyze resistance genes that are contributing to antibiotic resistance among certain bacteria, such as Staphylococcus aureus, having the gene MEK-A and being resistant to methicillin, or vancomycin-resistant Enterococcus, or multi-drug-resistant Mycobacterium tuberculosis. We also want to do genotyping studies on organisms, classification studies. This is usually done on 16S ribosomal RNA, and also in the discovery of new microorganisms. Whenever we're talking about clinical specimens, we have to discuss appropriate collection protocols. So when we're talking about microorganisms in molecular testing, we want to be able to collect the specimen in a way that's going to preserve the viability. So the organism has to possibly, stay alive, and also to keep the nucleic acid integrity of these microorganisms. We want to avoid contamination. The appropriate time, so if a specimen needs to be delivered to the lab within an hour, that has to be adhered to. Also, collecting the specimen from the appropriate site. Should it be blood? Should it be urine? Should it be another type of specimen? Using the proper equipment, so if you're drawing blood, what type of anticoagulants should be used, because some are inhibitors for certain molecular reactions. Should you use a wood or plastic applicators? There are commercially available collection kits that are commonly used, and whenever you're not sure about how a specimen should be collected before running a test, you need to look at the Clinical and Laboratory Standards Institute, or CLSI, guidelines. Preparing specimens, you always have to consider the specimen type. Is it stool? Is it plasmid? Is it spinal fluid? Is it something that needs modification? Depending on the microorganism that you're looking for, you may have to have more rigorous lysis procedures. So bacteria, especially certain bacteria, have thick cell walls. Your gram-positive organisms have a thicker cell wall than gram-negatives. So some organisms need a stronger lysis than your typical eukaryotic cell. You have to consider the number of organisms in a sample. So some of your specimens you're going to want to centrifuge in order to concentrate the bacterial cells or microorganisms in the specimen. You may need to inactivate any inhibitors that might be present, such as acid polysaccharides in sputum or polymerase inhibitors in spinal fluid. You need to inactivate your RNases if you're going to analyze RNA. We also have to consider quality control. So remember, our amplification methods, such as polymerase chain reaction, are very sensitive and very specific and also highly susceptible to contamination. So we have to make sure we control for all of these things. So in order to accurately interpret any tests in the laboratory, as you all know, we need to have proper controls. So we always have a positive control, which contains your template that will amplify. So you want to be sure that you are getting amplification or you're getting a positive test reaction so that you know that your kit is working properly. In your positive controls, you also might have sensitivity controls. There may be a positive control at the lower limit and a positive control at the upper limit of detection. You always want a negative control so you know that there's no contamination present. You will also want to have an amplification control, and this is an omnipresent control. So your template is usually going to be a housekeeping gene that's not related at all to your target sequence. And your housekeeping genes typically in prokaryotes are GRO-EL, RPOB, REC-A, and GYRB. So remember, these housekeeping genes are constitutively expressed. So they're on all the time. They are expressed at the same levels. So they're very good to use as controls for numerous types of amplification reactions. And remember, you always want a reagent blank that has no template at all, so you know if your sample has been contaminated with some other DNA. Usually, most methods in a clinical laboratory are commercial kits, and these kits commonly have internal controls. And these are amplification controls that monitor specific steps throughout an amplification method. So you might have a homologous intrinsic control, a heterologous extrinsic control, and a heterologous intrinsic control. So your homologous intrinsic control is a wild type derived control with a non-target derived sequence insert. It's added to every single sample after you extract your nucleic acids, but before you amplify. And amplification occurs of this non-target sequence using the same primers as for the target sequence. In the heterologous extrinsic control, again, it's a non-target derived control. It's added to every sample before you extract your nucleic acids. This control ensures that your extraction and amplification procedures were acceptable. In the heterologous extrinsic control, you need to use a second set of primers in order to get the control to amplify. So the amplification of the control can't interfere with the amplification of the target, or else this control is not going to work. Your heterologous intrinsic control, usually these are eukaryotic genes. So this serves to ensure that the human nucleic acid is present in the sample in addition to controlling for your extraction and your amplification. So this control is only going to amplify if you're using multiplex amplification, so that would be using several different primers, or using two different amplification reactions. So two separate tubes, one with your eukaryotic genes and one with your sample. So This is just an example of each of these internal controls. Your homologous extrinsic control is a control for amplification. Your heterologous extrinsic control controls for your extraction and your amplification, and your heterologous intrinsic control is your human gene control. So for your QC, you always have to consider false positives, and false positives commonly are due to contamination. So that's why you always want to have your blank, your no template at all. So if that gives you a result, you know that there's been contamination. Also, keep in mind that dead or dying organisms still have nucleic acid in them. So you are going to get a positive test reaction when you have dead organisms. So if someone has undergone antimicrobial therapy, you may get a positive for a specific organism, which would indicate that they have an infection, when in fact they just have dead organisms there. So you want to retest that specimen 3 to 6 weeks after antimicrobial therapy. So you want to collect another specimen from that individual 3 to 6 weeks after their therapy and then see if they have any positives. Because at that point, the organism should be dead and there should no longer be any organism present. Also, detection of less than clinically significant levels. So you may get a positive, but the person may only actually have one organism in them. Because remember, you're amplifying usually for these tests. So they may not actually have enough organism where it would be clinically significant. So we all harbor organisms in our body. So just because you're positive does not mean that that's the organism that's giving you symptoms. Again, quality control, we want to consider our false negatives. So the organism may actually be there, but the test result is negative. This could be due to improper collection or specimen handling, degraded nucleic acid. There might be inhibitors in the sample, so hemoglobin, lactoferrin, heparin, polyamines, all of these can inhibit amplification. You might have had extraction or amplification failure, which is why you really need those internal controls present, so you want to check those. And there could possibly have been technical difficulties with your instrumentation. Bacterial targets. The specificity of molecular methods targeting the primary nucleotide sequence depends heavily on the primers or the probes, depending on which detection system you're using. Also, choosing a target sequence is critical for the specificity of a molecular test. Many microorganisms share the same sequences in evolutionarily conserved genes. So that's something that always has to be kept in mind. So if you had a conserved gene among numerous different microorganisms, you wouldn't want to use that sequence in order to detect a specific strain. You want to use sequences usually that are unique to the target organism, so that if you get a positive result, you know it must be that specific organism that's causing the infection. So many tests I already stated used in molecular micro laboratories are commercially designed and FDA approved. Examples of FDA approved molecular microbiology tests. There are several different companies that have tests for chlamydia trachomatis as well as Neisseria gonorrhea. GenProbe has an FDA approved test for Mycobacterium tuberculosis, and there are some companies that have tests for HPV, CMV, as well as Group A strep. So there are other molecular tests available, but some of them have not been FDA approved yet. Different companies have used different methods in their tests. So you can see there's a PCR-based method for chlamydia, a ligase chain reaction, a TMA, a hybrid capture, and we discussed all of these different types of amplification methods in our amplification lecture. So at this point, we're going to be talking about specific types of diseases and molecular detection of different diseases. So now we are going to be discussing some of the methodologies used to detect these diseases, and we already discussed the details. So you may have to go back into your notes if you don't remember some specifics about some of the tests that we're going to talk about. Your target genes for a molecular assay are the genes that have been the targets for the design of primers, and these usually include ribosomal RNA, both in prokaryotes, the 16S and 23S. So ribosomal RNA tends to be a very common target, but there are other targets as well, depending on the microorganism that you're testing. So the 16S ribosomal RNA is the small subunit of the prokaryotic ribosome. 16S ribosomal RNA is commonly used to determine evolutionary and genetic relatedness of microbes. And many times, especially recently, a lot of genera and species names of bacteria have been changed, and it was because after analyzing the 16S ribosomal RNA sequences, they determined that two organisms that used to be in the same genus are now very different and should be in separate genera, or two organisms are very similar, so they're now in the same genera, etc. The 23S ribosomal RNA is the large subunit of the prokaryotic ribosome. Target genes tend to commonly be housekeeping genes that are always present and always expressed in your organism. But keep in mind, for diagnostic purposes, sometimes these housekeeping genes are your control genes, and you want a unique sequence for your molecular diagnostics, so that you can identify a specific organism. So it really depends on the type of detection and the method that you're using. So we're going to start off by talking about some respiratory tract pathogens. So respiratory diseases are significant cause of morbidity and mortality. There are numerous non-molecular detection methods. However, a lot of these non-molecular methods lack sensitivity, or they're very time consuming. So when we think about some of our respiratory tract pathogens like Mycobacterium tuberculosis, this organism takes 2 to 3 weeks to grow. So if you're trying to culture it in the laboratory in order to say that someone's infected, you really want someone to be diagnosed before 3 weeks. So that's why these molecular detection methods are very important. So molecular based assays, you want them to be able to detect and identify pathogens directly in the respiratory specimen. You don't want to have to grow up your mycobacterium for 3 weeks before you can perform an assay on it. Bordetella pertussis, Legionella pneumophila, these are very fastidious organisms that require special media to grow. So once again, a fastidious organism, it would just be easier to take the specimen, the respiratory specimen, do a molecular-based assay on it, and detect these organisms instead of having to grow them up on special media and special conditions. Chlamydophila, talking about one of these organisms that recently had a name change, used to be Chlamydia pneumoniae. It's now Chlamydophila pneumoniae. That organism is an obligately intracellular organism. It needs to grow within cells. So you can't just streak it out on a plate and stick it in the incubator. So this is another one of these organisms that it's perfect for molecular-based assays because you don't have to grow the organism up first. Mycobacterium tuberculosis. Usually, you take a respiratory specimen such as sputum and you make a smear and you look for the bacillus in the smear. Also, you're going to streak it out on plates that are specific for mycobacterium or in broth and grow the organism out. The sensitivity of smears can be between 22% and 80%. Cultures are more sensitive than smears, but Mycobacterium tuberculosis takes 2 to 3 weeks to grow in culture. There are liquid detection systems for Mycobacterium that you can now detect the organism within a few days. So those have improved diagnostics for Mycobacterium tuberculosis. GenProbe, in the last, I'd say, 10 years or so, had developed a DNA probe assay. The sensitivity, though, in some of the original assays were not good enough to detect mycobacteria directly from a clinical specimen. So nucleic acid-based testing allows Mycobacterium tuberculosis to be detected directly. So there are several different types of molecular-based tests that are now available. So there's the Amplified M Tuberculosis Direct test made by GenProbe. This is a transcription-mediated amplification which amplifies the 16S ribosomal RNA. And amplification and detection can be performed in 3.5 hours and has 100% specificity for members of the Mycobacterium tuberculosis complex. There's also the AmpliCore Mycobacterium tuberculosis PCR test, and this is a kit-based PCR assay. Your cells are lysed in the clinical specimen to release your mycobacterial DNA. The DNA is then denatured. Primers that are complementary to a 584 base pair region of the 16S ribosomal RNA hybridizes to your target sequence so that you get your amplification. A DNA probe specific for Mycobacterium tuberculosis complex is used, and this takes 6.5 to 8 hours and has a sensitivity of 55.3% in smear negative samples, but it has a sensitivity of 94.7% in smear positive samples. So another respiratory pathogen, Bordetella pertussis, is the causative agent of whooping cough. Children between the ages of 1 and 5 are the primary individuals who get whooping cough. The problem with Bordetella pertussis is there's a lack of a reliable culture method because the organism is highly fastidious. It dies very rapidly. It needs very specialized media to grow. So for Bordetella pertussis, the target sequences for molecular-based assays are located in the IS, which stands for insertion sequence 481. So this IS 481 insertion sequence is one of the target sequences. Others are the pertussis toxin gene promoter region, the adenylate cyclase gene, and species-specific porine protein structural genes. So molecular-based assays have been shown to have significantly higher sensitivities for diagnosing Bordetella pertussis than your typical culture traditional methodologies. So the IS481 sequence, this insertion sequence, that is used as a target to identify Bordetella pertussis. However, this same insertion sequence is found in another species of Bordetella, Bordetella homesii. This can give false positive results. So if you were to get a positive with the insertion sequence 481 and you wanted to be sure that it's Bordetella pertussis and not the other species of Bordetella, you can use the insertion sequence 1001 to discriminate between the two, because the 1001 is found in Bordetella homesii, but not in Bordetella pertussis. The insertion sequence 1001 is the target sequence for another species of Bordetella, Bordetella parapertussis. So in order to accurately detect Bordetella pertussis, you must amplify both the IS481 and the IS1001 targets. So you want to use both amplifications to enhance the specificity of your testing. So we're going to move on to urogenital tract pathogens. And the two urogenital tract pathogens where there already are numerous nucleic acid based tests are for Neisseria gonorrhea and Chlamydia trachomatis. Commonly, both of these are found in co-infections. So it's really nice to have a molecular test where you can test a clinical specimen and you can differentiate between both of these organisms and be able to identify both organisms in one specimen. So there are numerous nucleic acid amplification assays that are currently available. There are target amplification assays, and these are the Cobas Amplicore, the BD Probe Tech ET, and Aptima combo, or signal amplification assays, and that is a rapid capture system by Digene. So these assays can be performed in urethral or cervical swabs as well as urine, and in some cases on thin prep transport vials. The sensitivities range between 93 and 100%, and specificities range between 99 and 100%. So the only situation, though, where molecular-based tests are not acceptable for the detection of Neisseria gonorrhea and Chlamydia trachomatis is in the workup of children in suspected child abuse cases. So in these cases, you either have to perform a culture alone or molecular-based tests in addition to culture. Another issue when you're diagnosing and testing Neisseria and Chlamydia is test of cure. So with your molecular-based testing, you are usually amplifying nucleic acids, and you have nucleic acids present whether the organism is alive or dead. So if you find a positive, is it because the person is currently infected or because they've been treated recently and they have dead organisms present? So if you want to determine the efficacy of a person's therapy, you need to wait 3 to 4 weeks after treatment to do your molecular-based testing. Another molecular-based assay is for identifying antimicrobial resistance. So there are different types of antimicrobial agents used for treatment for microorganism or infectious diseases. There are antimicrobial agents that are static, bacteriostatic. These inhibit growth, and there are bactericidal or cidal agents that actually kill the organism. So antimicrobial agents are classified by the fact that they're either static or cidal, their mode of action, as well as their chemical structure. So your sites of action of your typical antimicrobial agents, there's various sites of action, and different antimicrobials will attack different parts of your microorganisms. So one type of antimicrobial agent is going to attack protein synthesis, so that the bacterial cell will not be able to make their proteins. Metabolism of nucleic acids, membrane integrity, so attacking the cell wall, etc. The mechanisms for developing resistance to antimicrobial agents. So your microorganisms have developed numerous mechanisms to resist being killed by these antimicrobials. These are enzymatic inactivation, altering the target so that the antimicrobial agent won't be able to bind and kill, altering the transport of the agent in or out of the cell, and acquiring genetic factors from other organisms that are resistant. So, in order to use molecular methods to detect resistance, we're going to talk about a couple of common resistant organisms. So, methicillin resistant Staphylococcus aureus, or MRSA, is caused by an altered penicillin binding protein, and this penicillin binding protein is encoded by the MECA gene. So you can rapidly identify MRSA isolates in a clinical specimen by direct detection of the MECA gene. And this molecular method is very critical for effective patient management. So there are polymerase chain reaction and other amplification-based methods that are commonly used to identify the MECA gene. Another organism that is highly resistant is vancomycin resistant enterococcus. So you can do polymerase chain reaction and other types of amplification to detect the VAN A, VAN B, and VAN C1 and C2 genes to screen for the presence of vancomycin resistant enterococcus. This is commonly done in fecal samples. Another concern is Staphylococcus aureus that is resistant to vancomycin, especially if we have our MRSA, methicillin resistant Staph aureus, that then become resistant to vancomycin, then there's not that many treatment choices currently available. Another concern is multi-drug resistant tuberculosis, and there have been many nucleic acid tests developed to directly detect mutations in genes that are associated with antimicrobial resistance. In an MDR-TB, the most common tests are to look at the CAT-G and INHA genes, which are associated with isoniazid resistance, and the RPOB genes, which is associated with rifampin resistance. And although all Mycobacterium tuberculosis contains these genes, specific mutations within these genes is what leads to being resistant to either isoniazid or rifampin. So the advantages of molecular detection of resistance to antimicrobial agents. Finding these genes or mutations in these genes is very strong evidence of resistance. So you can grow the organism up on a plate, okay, you know you have the organism. Or you can look for a target gene and know you have the organism using molecular-based studies, but is that organism resistant or susceptible? So traditional methods are performing minimum inhibitory concentrations to determine which antibiotics are going to be the most appropriate to get rid of this organism. But if you know that the organism has a specific gene or mutation, you know it's going to be resistant to specific antibiotics, so you don't even have to test for those antibiotics. You can do rapid detection without having to culture and grow up the organism, especially for those slow-growing organisms. You can also use molecular detection for direct comparison of multiple isolates in an epidemiological investigation. So let's say that there is a hospital study going on because numerous people have come down with a nosocomial infection with a drug-resistant organism. You can then use your molecular methods and test all of these different isolates from different individuals and see if they all have the gene or the mutation. Which brings us to epidemiologic studies. So when the same organism is isolated multiple times, it's important to determine if these different isolates are the same strain. So did these isolates come from the same source? Knowing these things are going to improve treatment and control strategies. You also want to do epidemiological studies for phenotypical analyses, such as antimicrobial susceptibilities, like we just talked about, or for genotypic analyses. You can do plasmid analysis, restriction endonuclease mapping, fingerprinting, or genotyping. So some terms that are commonly used for epidemiology. So we have an epidemic, which is a rapidly spreading outbreak of an infectious disease. A pandemic is a disease that sweeps across a wide geographical area, and epidemiology is a collection and analysis of environmental, microbiological, and clinical data. So in molecular epidemiology, we can do phenotypic analysis measures of biological characteristics of organisms. We can do molecular epidemiology for genotypic analysis targeting genomic or plasmid DNA to determine a specific species, a strain, or a type-specific DNA sequence. Some other definitions: an isolate is a population of cells in pure culture derived from a single colony. A strain is an isolate exhibiting phenotypic and or genotypic traits belonging to the same lineage. A clone is an isolate descending from a common precursor strain. A type is a specific and discrete unit of information or character belonging to a strain. And an outbreak is an acute appearance of a cluster of an illness caused by a pathogen that occurs in numbers that are in excess of what is expected for that time and place. And an endemic is occurring in a population with no deviation from the expected fluctuation in the number of cases. Okay. Pulse field gel electrophoresis. Now, we talked about pulse field gel electrophoresis way back when we were discussing our all of our different electrophoresis and visualization methodologies. Pulse field gel electrophoresis is a common electrophoresis method used in molecular epidemiology. This electrophoresis can distinguish organisms that have large genomes or multiple chromosomes. And if you want to review pulse field gel electrophoresis, that was back in Chapter 5. There is a national pulse field gel electrophoresis database stored at the Centers for Disease Control. Now, the pulse field gel electrophoresis, if you remember, this is a very labor-intensive procedure. So this is not a protocol that's going to be done in your typical clinical microbiology laboratory. Pulse field gel electrophoresis is done at health departments that are doing extensive epidemiological analyses as well as in research laboratories. You can use pulse field gel electrophoresis, though, if there were an outbreak to do strain typing. So in this example, you have your marker, you have your outbreak strain, and you have 6 isolates. And you're trying to determine if these 6 isolates of this organism are the same strain type as your outbreak strain. So you can see it using your pulse field gel electrophoresis that isolates 3 and 5 are the exact same strain as your outbreak strain, but where the pink circles are showing variation from your outbreak strain. So pulse field gel electrophoresis is very good at showing even minor variations from the outbreak strain, such as isolate 6. That, if you were to quickly look at it, it almost looks like it has the identical type or pattern as the outbreak strain, but that second band is a little higher up, so it is slightly different. Here it is again. You have your outbreak strain and your other strains, and you can actually see in the bottom gel, this is an actual pulse field gel electrophoresis as opposed to a schematic. And you could see these are CNS isolates, and you can look at the variations in the strain types based on the banding patterns. So criteria for pulse field gel electrophoresis pattern interpretation, and this is known as the rule of 3. So if you have no differences in your banding, the location sizes of your bands on a gel, pulse field electrophoresis gel, that would be interpreted as the test isolate is the same strain. So this strain is the same as the outbreak strain. In order for there to be absolutely no differences, there are no genetic differences between the two isolates, so they're indistinguishable. If there are 2 to 3 differences in the banding pattern, that would indicate that the test isolate is closely related to the outbreak strain. It may have one genetic difference to the outbreak strain. So remember, there are mutations that are occurring all the time. So it may have generated a mutation, and it is a closely related strain. If the fragment differences compared to your isolate and your outbreak strain, there are 4 to 6 differences, then it's a little bit iffy. It might be related to the outbreak strain. It may have 2 genetic differences, and it's possibly related. If there are more than 6 fragment differences, then this test isolate is unrelated to the outbreak isolate, and it would have greater than or equal to 3 genetic differences. You can also do your arbitrarily primed PCR, rapid amplification of polymorphic DNA, and that's abbreviated as RAPID, to compare your marker, your outbreak strain, and your isolates. And in this instance, all of the isolates are the same as the outbreak strain except for the 4 isolates that are indicated with the red arrow. And you can see the different banding pattern in those isolates. You can also do interspersed repetitive elements. So enterobacterial repetitive intergenic consensus sequences, or ERIC. These are 126 base pair long genomic sequences that are found in some bacterial species and are highly conserved. So these sequences are located between genes in operons or upstream or downstream of single open reading frames. The ERIC sequences are flanked by inverted repeats that could form stem, loop, or cruciform structures in the DNA. So these sequences are found only in gram-negative organisms, so your Enterobacteriaceae family members. So this is an example of your interspersed repetitive elements. So you have your repetitive element sequence with your inverted repeat, and you have your ERIC sequence with the inverted repeat, and you can do your PCR amplification outward from your repetitive elements, which is going to give you strain-specific amplicons. So we have isolate A and isolate B. You can see the different amplicon for each, and then you could look at your unknown strain. So is your unknown strain the same strain as isolate A or the same strain as isolate B? Other types of genotypic methods used for strain typing of organisms in molecular epidemiology studies would be plasmid fingerprinting with restriction enzymes, restriction fragment length polymorphism analysis, amplified fragment length polymorphism, interspersed repetitive elements, which we just talked about, ribotyping, SPA typing, and multi-locus sequence typing. So plasmid analysis. This is doing what's called plasmid fingerprinting And what you do is you isolate and restriction map your bacterial plasmids. So some bacterial strains carry different plasmids, and these will have different phenotypes. So the plasmids are distinguished based on the fragment patterns after you cut the plasmids with your restriction enzymes. A lot of molecular epi studies are done on tuberculosis. So, Mycobacterium tuberculosis, there are a lot of genotyping and strain typing techniques done on this organism. So, common techniques are restriction fragment length polymorphism analysis, and this is based on your insertion sequence 6110. In this RFLP, you're going to compare your fingerprints among various strains to determine clonality. So you're trying to determine if your isolates with similar fingerprint patterns are similar or clonal strains to your outbreak strains. So we already said the RFLP is based on the insertion sequence 6110. There are other genotyping methods for Mycobacterium tuberculosis, such as MIRU, which is Mycobacterial interspersed repeat units, and these are repeats that vary in their length and their sequence. There's also Spoligotyping, which is the nickname for spacer oligonucleotyping. And in Spoligotyping, there are 10 to 50 copies of a 36 base pair direct repeat region separated by spacers, and the spacers have different sequences. So you can decipher your strains based on the presence and or absence of specific spacers. And spacer sequences are conserved among the strains. So in insertion sequence 6110 based genotyping, this is a typical southern blot technique. You isolate your DNA, you digest with the restriction enzyme PVU1. You use the insertion sequence 6110 probe to hybridize to your DNA. It's going to bind anywhere that to the right of where you cut with the PVU1 restriction enzyme. The size of each fragment depends on your distance between your PVU1 cut sites. In MIRU, this involves PCR amplification and gel electrophoresis to categorize the number and size of repeats in 12 independent loci of the Mycobacterium tuberculosis genome. So each loci has a unique repeated sequence, and the sizes of the loci A, B, C, and D in 3 different strains are shown in the following slide. So here we have your A, your B, your C, your D, and you could see the different sizes. So you have in one strain, BCG, you have your marker, and you could see your pattern at loci A, B, C, and D. For another strain of Mycobacterium tuberculosis, H37RV, you see they have a different genotyping, MIRU genotyping. So you know that those 2 strains are not the same. And then you have strain X and its fragment banding pattern at A, B, C, and D loci. So you can compare your unknown strains to known MIRU genotypes to determine what strain you might have. Spoligotyping is looking at the direct repeat locus or DR locus, and this is a chromosomal region containing 10 to 50 copies of a 35 or 36 base pair direct repeat separated by a spacer with various sequences, and each is between 37 and 41 base pairs. The insertion sequence 6110, a copy of that is inserted within the 36 base pair direct repeat in the middle of the DR locus. So Mycobacterium tuberculosis have some arrangement of spacers, but they differ in the terms of the presence or absence of specific spacers. So this technique involves PCR amplification of the DR locus, followed by hybridization of labeled PCR products to a membrane containing oligos or small little pieces of DNA corresponding to each of the 43 spacers. So this is just a schematic of your Spoligotyping, where you have your insertion sequence 6110 that's in the middle of the DR locus. You have your direct repeats and you have spacers that vary in their sequence, and you're just going to do an amplification and blotting technique to determine if you have the presence or absence of a specific spacer. You can use these various techniques to do dendrograms to determine genetic relationships of various Mycobacterium tuberculosis organisms. Why do we want to use genotyping for Mycobacterium tuberculosis? It's going to confirm the occurrence of cross-contamination in the lab to eliminate false positive results. It's going to permit evaluation of isolates with different patterns of drug susceptibility to aid in determining the best treatment strategies. It can be used to evaluate second episodes of tuberculosis. So someone had tuberculosis and then they come back several years later and have tuberculosis after treatment. Is the second episode a new infection with a different strain, or is it due to a relapse of the original infection? Also, to evaluate outbreaks. So you want to do genotyping and molecular epidemiology to determine if an outbreak has occurred or if there's just an increase in TB cases in that region. Comparison of your molecular epidemiology methods. So here you have your various methods, the typing capacity, the discriminatory power, the reproducibility, the ease of use, and the ease of interpretation. So viruses, classical methods to detect viruses include antibody detection, antigen detection, or culture. Molecular methods of detection include target, probe, and signal amplification methodologies. So tests are designed for identification of virus, to determine viral load or the number of viruses per ml of fluid, as well as genotyping by sequence analysis. So we're going to start off talking about human immunodeficiency virus, and this is a retrovirus that makes a DNA copy of genomic RNA using a virally encoded reverse transcriptase enzyme. Diagnosis of HIV is through detecting antibodies specific for HIV. Now, nucleic acid-based tests are performed on HIV patients to determine how actively the virus is replicating, and this is done through viral load studies. Also, to determine when to start antiretroviral therapy and to monitor the efficacy of treatment. So to determine viral load, so the amount of HIV that should be, or viral load, is used as a marker for disease prognosis. Patients are 10 times more likely to progress to AIDS within 5 years if they have viral loads above 100,000 copies per mil within 6 months of seroconversion. So the aim of antiretroviral therapy is to get the viral load below 50 copies per mil of blood. Patients who maintain viral levels at less than 10,000 copies per mil in the early stages of infection are at decreased risk of progressing to AIDS. Viral load determinations should be made before therapy is started, then 2 to 8 weeks after therapy initiation, and then every 3 to 4 weeks thereafter, 3 to 4 months thereafter, to assess therapeutic effectiveness. So since nucleic acid amplification assays use different techniques, right? One person may use a probe amplification, one lab may use a signal amplification. The results from one method may not be comparable with another method. So in analyzing viral loads in a patient, the same method should be used. So the patient specimen should be sent to the same laboratory each time, and labs should notify. I don't know if this is done, but it would be nice if physicians who work with HIV patients would be notified if the methodology were to change, so that they would know that maybe these results aren't making sense because the method was changed. Lower limit of detection is defined as the lowest viral concentration that can be detected 95% of the time in the test assay. So in order to be clinically relevant, the viral load changes from one determination to another have to be at least threefold different. So currently available FDA-approved HIV tests include the Bayer-Versant HIV RNA assay, the Roche Amplicor Monitor test, the bioMérieux NucliSense HIV-1, and Abbott's Real-Time HIV-1. And these all use different methodologies: branch DNA, reverse transcriptase PCR, NASBA, and real-time PCR. So here is a chart showing the test performance features for viral load measurements. So the sensitivity, the accuracy, the precision, the specificity, the linearity, and the flexibility. You can use nucleic acid-based testing to genotype viruses, and this is used to monitor the development of antiretroviral drug resistance in HIV. Viral genes mutate to overcome the antiviral agents. So gene mutations are detected through sequencing methods. And mutations are divided into two groups. There's primary resistance mutations which affect drug sensitivity but may slow the viral growth. And there's secondary resistance mutations which compensate for the primary resistance growth defects. We're going to talk about hepatitis C virus, and diagnosis is the very similar approaches as for HIV. So there are various nucleic acid amplification assays that are currently available. Qualitative Hep C viral RNA assays are performed on patients with positive antibody results to confirm an active infection or for immunocompromised patients. Quantitative hepatitis C viral RNA assays are used to determine viral load and monitor viral replication in response to antiviral therapy. There are 6 genotypes of hepatitis C virus: 1A, 1B, 2, 3, 4, 5, 6, as well as 80 subtypes. So the genotype present in a given patient determines the treatment protocol that's used. So the hepatitis C viral genotype is determined by analyzing the 5' untranslated region, because this is the most highly conserved region of the Hep C genome. Patients who have viral loads greater than 2 million copies per ml of plasma are not going to respond as well to conventional interferon ribavirin therapy. Genotypes 2 and 3 will respond better to treatment than genotype 1. And, and a 2 log 10 decrease in hepatitis C viral RNA indicates that a patient will respond to treatment. So in summary, molecular-based methods offer sensitive and direct detection of microorganisms. Due to high sensitivity and specificity, proper quality control is critical for molecular testing. Several molecular methods are used to type bacterial strains in epidemiological investigations. Target, probe, or signal amplification procedures are also used to determine viral load. Now, just to end off, we are going to discuss a molecular epidemiology criminal case. So this is molecular evidence of an HIV-1 transmission criminal case, and this was published in PNAS in October of 2002. So this was one of the first criminal cases where a virus was used for a conviction. So a gastroenterologist was convicted of attempted second-degree murder by injecting his former girlfriend with blood or blood products obtained from an HIV type 1 infected patient under his care. So phylogenetic analyses of the HIV-1 sequences were admitted and used as evidence in this case, representing the first use of phylogenetic analyses in a criminal court case in the United States. So the charge was that on August 4th of 1994, a gastroenterologist made a mixture of blood products from two patients under the doctor's care, one infected with HIV and the other infected with hepatitis C, and he infected his former girlfriend by intramuscular injection. So the risk factors from 1984 to 1995, the victim reported having sexual contacts with 7 men, including the doctor, all of whom were interviewed by local law enforcement agents. The 7 men were tested between the years of 1995 and 1998 and were found to be negative for HIV-1. The victim was a nurse who had no documented reports of needle sticks, but did report in the mid-1980s that an HIV-1 infected patient splashed saliva onto her skin while spitting into a pan. The victim was tested after that incident and was found to be negative for HIV-1. The victim had donated blood to a local blood bank on several occasions, for which she was tested and found negative for HIV. This was done in October of 1992, May of 1993, and April of 1994. So in January of 1995, the victim tested positive for HIV-1. She then accused the physician ex-boyfriend of infecting her with HIV-1 from an intramuscular injection during an argument in early of August 1994. So to add fuel to the fire, law enforcement agents identified an HIV-1 infected patient whose blood had been drawn in the doctor's office on August 4th, 1994. The patient's name was one of the last recorded entries in a reportedly missing log notebook that was discovered after a search warrant had been obtained to search the physician's office. The patient's blood draw was recorded in a different manner from the usual operating procedures of the doctor's office, so it was a little skeptical. The patient was a homosexual male who was infected with HIV-1 in 1990. So controls. The most appropriate controls for the current study were HIV-1 infected individuals living in the same area. So 32 blood samples were obtained from available HIV-1 infected individuals from the local metropolitan area. These controls provide the greatest opportunity for identifying an alternative HIV-1 transmission between these individuals. The use of local sequences also allowed the consideration of the possibility that HIV-1 DNA sequences derived from the suspected transmission pair were representative of an HIV-1 strain ubiquitous to that area. So here's a summary of that control group. The risk factors involved, so 57% of the controls were homosexual, 18% bisexual, or heterosexual, 11% were blood transfused 7% were bisexual, 4% IV drug users, 4% had incidents involved with sharps, and as well as their dates infected. So, in order to reduce lab error, the experimental data were derived from two independent laboratories, first performed at Baylor College of Medicine and then confirmed at the University of Michigan. So the strategy of the independent testing in the two different laboratories from two separate blood draws, each for the patient and the victim, allowed the exclusion of the possibility that the relatedness of the HIV-1 DNA sequences derived from the patient and the victim were the result of either laboratory error, sample mix-up, or cross-contamination. In order to eliminate bias, the second study was conducted in a blinded fashion, so the identities of the samples were not revealed to the Michigan group until the analyses of both samples were completed. Both samples were analyzed at different times, separated by thorough cleaning of all laboratory equipment and surfaces, and in all instances, peripheral blood mononuclear cells were isolated from whole blood, the PCR was set up, genomic DNA and DNA sequences were each performed in physically separate laboratory areas. For the analyses, genomic DNA from each individual was used to PCR amplify an 858 base pair envelope gene fragment and a 1,147 base pair reverse transcriptase gene fragment. Phylogenetic analyses of the HIV-1 reverse transcriptase and envelope DNA sequences isolated from the victim, the patient, and a local population sample of HIV-1 positive individuals. So here's the phylogenetic analysis of the GP120 region of the HIV specimens. Here's the reverse transcriptase region. And for the results, the victim's HIV-1 sequences were found to be most closely related to and nested within a lineage comprised of the patient's HIV-1 sequences. So this finding was consistent with the direction of transmission from the patient to the victim. The analysis of the victim's viral reverse transcriptase sequences revealed genotypes consistent with known mutations that confer resistance to AZT and similar to those genotypes that were also found in the patient. So, in conclusion, the gastroenterologist was convicted of second-degree murder by injecting his former girlfriend with HIV, and this was all done through phylogenetic molecular-based analyses.